Hey everyone, so we're going to start chapter 3 with a short discussion of the PowerPoint introduction, and then we're going to get to handout 3A. So let's look at our PowerPoint, and the PowerPoint's going to be over cell structure and function. So if we look at this, I just want to remind you that I will post a study guide on D2L, and that will help you get ready for this first test. And so the first test will be over chapters 1, 2, and 3. So look for the study guide. I'll post it in chapter 3 of D2L, and I'll make that available for you. So this chapter is all about cell structure. We're going to have a short crash course in basic cellular biology. And so as we go through this, the cell, remember, is our basic unit of life. So the cell is our basic unit of life. It is made up of the plasma membrane, which is that barrier that is between the cell and its environment. And so let's get this going. So our plasma membrane is going to be a divider. We're going to talk about it first. Then we have the cytoplasm that's made up of a fluid and protein that supports the cell called the cytoskeleton. And then a bulk of this chapter is just going to be a review on the organelles. So we're going to go through the organelles that perform certain functions that most cells have. Okay, so to get started here, we're going to go to handout 3A. And this handout is going to give us a discussion and introduction of the plasma membrane. So I'm going to switch over to handout 3A and let's discuss it. So 3A is going to be kind of a crash course in the plasma membrane. Now, the plasma membrane is going to be made out of various biomolecules. So those biomolecules are primarily going to be a structure called a phospholipid. So our plasma membrane is made out of primarily phospholipids that form what we call a lipid bilayer because there's two layers. So as you see this, there's a layer on each side. So there's a layer pointing away from the cell to the outside world, and then a layer that's pointing inward towards all the cell components. Now, this layer is made up of a biomolecule called a phospholipid. And a phospholipid is made out of a water-loving component called a hydrophilic head. It's made of phosphorus. This head, represented kind of by these blue little spheres, like water. So there's a lot of water outside the cell and a lot of water inside the cell. So these hydrophilic water-loving heads are oriented towards the water. Then we've got this fat lipid tail that are hydrophobic. They don't like water, they're hydrophobic, and they orient themselves away from the water as much as possible, so they point inward. So we have a layer on each side with these hydrophobic tails pointing in. Now this is primarily what the plasma membrane is made out of. It also has some other components, and that's what this picture underneath it is showing. These other components are going to be things like proteins and fats, glycolipids, glycoproteins, cholesterol, throughout this phospholipid bilayer. So this second picture shows my phospholipid bilayer, but it shows all these other components within this plasma membrane. Now, what I want to do is talk a little bit about what these different molecules do. So it says a lot of these proteins act as channels to allow molecules in and out of the cell. So these act as like little pipes. So these little purple kind of sausage looking things are proteins we call channel proteins. The channel proteins move things in and out of the membrane. Some are identification markers. So some of these glycoproteins are sticking out of the plasma membrane, and they are used to identify the cell to, let's say, our immune system. Then we have cholesterol. Cholesterol is kind of scattered throughout our phospholipid membrane, and that's going to be there to stabilize the membrane's integrity. Kind of makes that membrane a little bit more rigid. We do say that the plasma membrane is a fluid mosaic model because those phospholipids can fold and roll and move over each other. The cholesterol kind of helps stabilize that, so these phospholipids can't move as much. That fluid mosaic model is a little bit like oil sitting on top of water. So the oil can flow and move and go over each other, under each other, and the cholesterol kind of stabilizes that. So if we look at this, let's first talk about these channel proteins. So there are different kinds of channel proteins. We have 3 types of channel proteins that allow things to move in and out of the cell. The first one is called a non-gated channel protein. They're like doors that are always open. So they're always open. Things can just move straight through unimpeded. Those are non-gated. Then we have ligand-gated channels. These are channels that have a door. So we can shut the door and only allow certain things to pass through the membrane. These gated channels have a gate or a door. They also need a key to open the door. And the key is a chemical called a neurotransmitter. So this neurotransmitter can come on to the channel protein, lock on it, open the door so that molecules can move through the membrane. We'll talk a lot more about neurotransmitters and ligand-gated protein channels in the next couple of chapters when we get into muscles and nerves and that kind of thing. We also have voltage-gated channels. Voltage-gated channels open due to an electrical charge. So imagine that we zap this membrane with electricity, and that can open up a membrane protein channel that allows molecules to move through. So this would be not like a locked door, but maybe like a door at the grocery store that senses you're there and then automatically opens if you are the correct molecule. So those are examples of channel proteins. Now, I also mentioned that we have identification markers, and these identification markers are located on the plasma membrane of cells to identify it for various purposes. Now, I've got 4 of them that are listed here. The first one is identification markers can ID the cell to our immune system. This is how our immune system recognizes us versus a foreign body that it wants to attack, like a bacteria. Bacteria have different identification markers. And so our immune system, the immune cells, our white blood cells, are going to be traveling through our body, and every time they come around a cell, they taste it. If it's us, we have matching identification markers. if the identification markers don't match, the immune system knows to attack that. So that's one example. Another example is our egg and sperm. So the egg cell has certain identification markers on its plasma membrane that the sperm can recognize as that egg. You see, the sperm are not so bright, and they are trying to just swim and, find that egg, but they don't know what the egg is until they find that identification marker. So once they taste that egg and they know that that's the egg, then they try and penetrate it to fertilize it. Other identification markers are on these channel proteins. So some of these channel proteins need an identification marker so that the neurotransmitter can recognize it. And so we've got identification markers on the channel proteins themselves. The neurotransmitter comes in, latches onto that identification marker, and it knows that it's at the correct spot. So it can open up that channel protein. The last example is identification markers are also hormone receptors. So our hormone receptors are going to detect what cell the hormone needs to affect. So I have handout 1B as an example, but handy, I've actually got handout 1C. So if you remember handout 1C, we were talking about this hormone called oxytocin. And oxytocin, I mentioned, is secreted out of the posterior pituitary gland. It's going to flow through the bloodstream to our uterus during childbirth. And oxytocin is going to tell that uterus to contract. Now, the big question is, how does it know? How does it know to affect the uterus and not some other organ? The answer to that is the uterus itself has identification markers on the uterine cells that act as hormone receptors. So the uterus, the muscles of the uterus have oxytocin hormone receptors. So when oxytocin comes in contact with those receptors, the oxytocin knows to affect that cell and cause those uterus muscle cells to contract. You see, the rest of our body doesn't have those hormone receptors. So for example, the stomach doesn't have oxytocin receptors. So when oxytocin is flowing through the bloodstream, it's not affecting our stomach or our esophagus or the pancreas or the liver, because those organs and tissues don't have the hormone receptor. The uterus, on the other hand, and our mammary glands, they have the oxytocin hormone receptor. So they are affected by oxytocin. We could use the same example for antidiuretic hormone. The kidney has hormone receptors on the kidney cells that affect or that cause the effect of antidiuretic hormone. Other cells don't have this. So we don't have antidiuretic hormone affecting other organs. So this is how a hormone knows what organ to affect. It's because those organs have these hormone receptors. So that's pretty cool, and that explains a lot that I didn't really cover in chapter 1, which is how do those hormones affect a certain organ and not something else? It's because of these hormone receptors on the plasma membrane. So this takes care of handout 3A, and next we'll continue on with the next video with more plasma membrane stuff. All right, I'll see you later.